Okay, so here I'm going to discuss how antigens and antibodies interact and how these interactions are used in antibody assays in order to determine, for example, if you have an immune response to a particular pathogen, and antibodies are good at identifying people who have been exposed to different pathogens. So first, let's just talk about antibodies and antigens. So we know that antigens are going to come in a wide variety of shapes. So we have on one end our antigens, and they have many shapes, sizes, and our antibody repertoire has to be able to recognize all of these different shapes and sizes. Some of them can be very simple, so we can have simple structures. We can have some that are going to be much larger epitopes. And remember, an epitope is just part of the antigen that is being recognized by the actual antibody. And then we can also have complex antigens. And so each of these are going to be bound to the antibody in different ways. So a simple structure is in essence going just to fit in a pocket. And we have the larger structures that are going to fit into an antibody groove. And then we have our complex antigens, which are going to have, in essence, an extended surface. When they interact with the antibody. And so you're going to have an infinite variety of binding sites for antigens in your antibody repertoire. And then you're going to use these antibodies in yourself to protect. And we are going to use these antibodies in the lab for diagnosis. And so the nice thing about antibodies is that they have very high specificity and they also have an affinity. So we've already talked about antibodies and how you have one antibody can only bind to one antigen, and that's the specificity part. And then the other part of it is how well can that antibody actually bind to that antigen, and that's the affinity part. And so the affinity and specificity are important not only for protection, but are also key in diagnosis because you have to have confidence that you are actually identifying an antibody for a particular pathogen. And so the affinity and specificity are what help to make that diagnostic possible with that. So the affinity and specificity, you can think of them similar to the way we think of other proteins in our body, protein interactions. So we have our enzymes that are very specific for their substrates. And so that's very similar to an antibody-antigen interaction. And so this extraordinary specificity and affinity for antibodies to their antigens is what is the basis for a lot of our diagnostic testing. Now let's just talk a little bit about our actual antigen binding sites. So our antigen binding sites are going to be based on whether you have a small antigen or if you have a larger antigen or if you have that extended surface antigen. But in essence, the antibody is going to bind to them at their antigen binding site, which is, again, we'll draw this lovely Y. So we have the antigen binding site here. And so if you just have a small antigen, that small antigen might just fit right into the pocket. So this is a pocket binding. And then if you have a larger antigen, this antigen might instead, sorry about that, fit into a groove. Make a different color. And so this would be a groove. And then an extended surface antigen would simply bind to the antibody, or an antibody would be able to bind to it that is able to bind to the extended surface. So it would be a much larger antibody binding groove. And here's our antigen that binds to that guy. And so this is an extended surface. But the basic story is that all antigens come in different shapes and sizes, and your antibodies have to be just as flexible and diverse in their development to bind to all these different shapes and sizes. Now, another thing that's important with respect to antibodies is cross-reactivity. And so an antibody can occasionally bind to more than one antigen, which is referred to as cross-reactivity, or multi-specificity. And so in this case, for example, you will have your antibody, and it will have on its surface an antigen binding site, and it can bind to an antigen on this binding site. And the antigen might be complex. And so your antibody is going to bind the immunogen, this antigen. Let's make it red. And then you can potentially have a cross-reactive antigen. So for example, we'll take this same antibody, and if you have an antigen that looks similar to that red antigen, but it's now a blue antigen, but it still has kind of that little dip, kind of still has that, but it's a different antigen altogether. This will be immunogen 2, and it looks similar enough to immunogen 1 that it can bind to the same antibody. So this is antibody 1, and this is also antibody 1. And so this is cross-reactivity. And so the antibody 1 is specific for antigen 1, but antigen 2 fits well enough into the same groove to create a stable binding interaction. And so this cross-reactivity can happen because of a sufficient number of interactions between the antigen and antibody that create this stable structure. And so it's important that cross-reactivity can also have clinical consequences. And it's thought that some cross-reactivity might help to explain some autoimmune diseases. Now, I just want to talk now about how antibodies are going to be used for diagnostic tests. So there are both qualitative and quantitative approaches to studying antibodies, or you can study antigens they target. One of the most common tests are used for diagnostic is the ELISA. ELISA is enzyme-linked immunoabsorbent assay. And so an ELISA is very sensitive and simple. It basically uses the approach that you are going to have a particular antibody that's specific for a particular antigen. And so you have what we have plates, and the plates have these wells. So this is a well. And you basically are going to coat the well with your antigen. And so you're going to put the antigen on the bottom. And this antigen, for example, might be influenza H protein, hemagglutinin. And so you coat the well with the antigen. And then the next step is that you're going to add your antibody. And so, for example, you can have several wells. Typically, these are done in very large plates. You can get 96 well plates, 384 well plates, so you can do a lot of different replicates. But in essence, you have your antibodies. So you'll have your different patients. So you'll have patient 1, 2, 3, and 4. And you're going to add the antibodies to this well. And so this time, your trunk's going to be faced up, and then you're going to have your Ys out. And they're going to see if they bind specifically to your red antigen. We have our person 2 that has those red antibodies. Patient 4 will also have those red antibodies. And patient 3 and patient 1 do not. And so basically the antibodies will bind, and then you're going to have a step where you wash the plate. And after you wash the plate and you add a reagents to detect binding of your antigen and antibody, basically what happens is the purple antibody and the orange antibody do not stick. The red antibodies will stick, and you add a fluorescent marker, a tag. And so what you basically get is you get a color change if there's positive. And then in patient 1 and 3, there is no color. So you know that patient 2 and patient 4 have antibodies that bind to influenza hemagglutinin protein. And you know that patient 1 and patient 3 do not have these antibodies. And so basically an ELISA is a pretty easy way, as long as you know, you have to know the target antigen. So remember, this is very specific. So if you were just to coat this with influenza virus, whole virus, you might get binding patient 1, 2, and 3, and 4. You might not. But you wouldn't know what protein they were actually binding to. You wouldn't know if they're binding to the hemagglutinin. You wouldn't know if they're binding to neuraminidase. And there's a lot of cross-reactivity in these culture plates especially. There's a lot of background. So it's very important to have a very specific antigen that you're looking for a very specific antibody response to when you're doing an ELISA. Okay, so there's also other tests you can do. There's one particular that's used for syphilis. It's a serologic test. And so this is serology, which is serum, and we're looking for antibodies. So we have a spirochete, Treponema pallidum, which causes syphilis. So you have a spirochete bacteria. And basically the bacteria is a rod, and you can use a fluorescent antibody absorb test called FTA-ABS. And this is a fluorescent treponemal antibody absorbent test. Okay, and so this is where you take serum and you have your antibodies that are going to be mixed with the treponema pathogen, the spirochete. So you have, again, you have patient 1, you have patient 2, you have patient 3, and this time in your wells you're going to, for example, or you're actually going to use a slide. So this is a slide. You're going to have your treponema bacteria on your slide. Now, you wouldn't see them as being green. I'm just showing it as being green, just so you can see it. And then what you do, let me split these guys up. What you do is you take serum from each of the patients and you add it to the slide. And then you are going to add a second conjugated antibody to bind to the humans. So remember, when we talk about antibodies, we talk about them in the shape of a Y. And remember, this is where your spirochete is going to bind if it's specific. I'm just going to draw it as a little spirochete. So here's your bacteria. And that's just an epitope of the bacteria. The bacteria will, of course, be larger, but I'm not drawing that. And then you're going to have in your trunk, this is the FC region, and this is coming from people. This is patients. These are people. So this is a human antibody. And so what we do, which is really clever, is we take our human FC region and we inject it into an animal, such as a rabbit. And then the rabbit will make I'll just draw it out, anti-human FC. Okay? Then you can take this anti-human FC and you can tag it. And you can tag it with a fluorescence. That could be able to fit fluorescence in there. So you can make a tag that will make it glow if it binds. And so when you add patient 1, patient 2, patient 3, you are going to follow this. So this is step 1. And step 2 will be the anti-human tagged with our fluorescent marker. Okay, so Patient 1 has these purple antibodies that do not bind. Patient 2 has these green antibodies. That bind, and patient 3 will have green antibodies that bind. And so you're going to get your fluorescence tag on patient 3 and patient 2, but not on patient 1. And so patient 2 and patient 3, your treponemin will light up under a fluorescent microscope. But your patient 1, since it does not have any antibodies, will not light up. And so that's basically the way that this test for syphilis is done. Now there's also lateral flow tests. And so lateral flow tests are a way to measure your antibodies, proteins, or antigens in your body fluid using a very simple, straightforward ELISA-like test. And this is, for example, with how they make pregnancy tests. Now, the basis of this is to determine if a specific protein, antigen, or antibody is present in whatever body fluid you're testing. You can be testing serum, you can be testing urine, you can be testing saliva. And it's very similar to an ELISA, except for it's engineered to flow. So instead of doing it in those little wells, you're going to do it on some sort of slide or a mechanism that allows you to first do the binding part and then move to the detecting part and then move to the results part. So they're usually simple to perform and they're relatively cheap. And so the pregnancy test, for example, is going to confirm whether or not in the urine of a potential pregnant woman there's the presence or absence of human chorionic gonadotropin hormone. So that is what the pregnancy test is testing for. And so basically you can think of it as a slide approach, even though you don't see any of this. This is what it is. So you bind, for example, on the surface of the slide, you're going to bind antibodies for human gonadotropin, choriogenic gonadotropin hormone. So these will be okay, so you have your urine. This can be placed on this strip, and it's made up of like an absorbent paper-like material. And the urine sample at one end of the stick will be drawn along by a paper-like capillary approach. And during this time, it's going to encounter these antibodies that are going to be conjugated to a microbead. And this microbead is what's going to actually bind the human chorionic gonadotropin hormone. So you take your serum, you have your blue, and then you add your serum across this, or urine, if it's a pregnancy test. And then basically, when the urine mixes with this, if there's any of the hormone present, the hormone is going to bind to the antibody bead complex. So we'll add the hormone as these pink dots. So here's our hormone. It starts to bind to these bead. And so here's our human. So now they're bound. And then the urine antibody mixture next reaches an area where the second antibody against a different epitope of the hormone has been also put on the surface. So we're going to have a next layer. I'll kind of draw it here. And here you're going to have another antibody. And this is actually going to be against the hormone. And this antibody will bind to any hormone-bound antibody B complexes. And so this is what it's going to look like. It'll bind to your hormone attached to your bead. And there's your bead, which is bound to that antibody. Typically they'll use a mouse antibody or something like that. And then if you have sufficient hormone present, you will get a colored line when the beads and the particles aggregate. So the beads and the particles will continue to flow. So here's the flow. And they'll continue to flow. And then you'll get an aggregation. Which aggregation just means a lot together. Just draw some of them. So they're kind of clumping. And then we have our hormone in here attached to our beads in here. And this aggregation generates, in the case of a pregnancy test, a line. Now the line is coming from the first hormone that actually has the indicator on it. So this antibody for this has the indicator. Okay, so these are relatively simple, relatively cheap, and they're based again on the specificity. In this case, the specificity is for a hormone. And you're kind of doing it double. So you have the first binding of the hormone to the first antibody, which has a bead bound to it. And then you're moving to a second antibody that in order for it to clump together and generate the visual line, it has to have enough aggregates of that hormone bead complex bound to those second antibodies. And then you get the visual positive test. Now there's also immunofluorescence. And so immunofluorescence is where you have proteins and you have an antibody that binds to them, and then they're able to fluoresce under UV light. So in this case, again, you're using antigens. And then you're going to bind the antigens with some sort of antibody. Actually move this. So this is one arm of our antibody. And over here will be the other arm. And so here's our antibody. Here's our FC region. And so your antibodies will have been covalently attached to some sort of fluorescent compound, which are called fluorochromes. Oops, let's make it. Now, a fluorochrome can be many different things. One that's widely used is called FITC, fluorescein isothiocyanate. And this is going to be able to couple or bind to free amino acid groups on proteins. And so basically, the antibody bound to the fluorochrome is mixed with whatever you're trying to detect, and when the antibody binds, it will fluoresce again. This is just another way to make it fluoresce. Now, the fluorescent microscopes will be using UV sources, and these are going to cause excitation in these different fluorophores or fluorochromes, and you're going to be able to have multiple fluorochromes due to different excitation when they're exposed to the immunofluorescence wavelengths. This test is used to detect antigens in cells or tissue sections, and it can also screen for autoantibodies on a cell or tissue. So, for example, if you take a liver biopsy, you can basically cut the liver, the hepatocytes, and you can put them on a slide. So here's our hepatocyte. And these are very thin sections, but you can still see the structure of your hepatocyte, the cell. And let's say that you know that the hepatocyte has an insulin receptor on its surface. So you can generate an antibody, an anti-insulin receptor antibody. And so you would make this antibody. I did that wrong. And so this would bind to insulin receptor, so I'm putting an I there. And then this antibody will be tagged with a fluorescent marker. Let's say it's green. You basically add this antibody to your slide. Oops, that didn't work. And then it'll bind. So it'll bind to the actual tissue. And you'll get like these little green, sections, the edges, for example, because the insulin receptor will be on the edges. And so that the antibody is basically glowing wherever the insulin receptors are. And so you can do this for a multitude of different proteins you're interested in on the hepatocyte. You can look at glucagon receptors. You can look at different enzymes, different proteins that are specific for the hepatocytes. And all you have to have is an antibody tagged with a fluorescent marker to see them. And the nice thing about this is you see the structure. So when we did our ELISA, you just got a positive or negative, a color or no color. Here, you're actually seeing where the fluorescence is. So that's the nice thing about using immunofluorescence. And so we also have what we call FACS analysis, flow cytometry. This is what I call the workhorse of the immunologist. Now, flow cytometry, there's many different things you can watch and see. You can see the machine actually work. But in essence, flow cytometry, what it allows you to see is it allows you to see differences at the level of a single cell. Now, when we talked about the tissue on the slide, you're looking potentially, I just showed you a hepatocyte, but in general, you'll see a whole liver on the tissue. And so what flow cytometry does is it takes those cells, separates them, and you can start to see differences in them. So, for example, with flow cytometry, again, it all goes back to antibodies. So we're going to have an antibody that targets CD4 cells. Okay, and we'll have an antibody that targets CD8 cells. And we'll have an antibody that targets B cells. That's CD19. And we'll have an antibody that targets macrophages, which is called CD11B, for example. We'll just start there. Okay, so all of these are antibodies, okay, nothing different. And for example, we'll make the CD4 cell will be green, we'll make the CD8 cell orange, We'll make our CD19 pink, and we'll make our CD11B blue. So because you can tag them with different fluorescent markers, you can actually differentiate the green CD4 from the orange CD8. And what basically happens is you would take all of these plus your blood cells and you incubate them, and then you run them on the machine, a flow cytometer. Okay? And so the machine is made up of a lot of different components. It has different lasers we're not going to go into. But in essence, what the machine is able to do is able to show you how many CD4 T cells are in this blood sample, how many CD8s, how many B cells, how many macrophages. And so it will look like something like this. You'll have a graph. It always gives you graphs. And the first graph, you're going to get a side scatter by a forward scatter. And forward scatter is size, and side scatter is complexity. And so we have here 3 lymphocytes, right? Here's a lymphocyte, CD4, CD8, and our B cell. And we have 1 macrophage. Macrophages are much bigger than our B cells and T cells. So in essence, what you would get is you would get a population that shows up here. There'll be a bunch of dots. Each dot resembles 1 cell. And this will be our lymphocytes. And then we'll get another population somewhere over here. And these will be our macrophages, because they are larger than lymphocytes and they are more complex. They have a lot of granules, they have a lot of things going on. Lymphocytes are just basically a big nucleus until they get activated, but they're typically much smaller and they don't have as much complexity as our macrophages. Now what you can do is you can take these populations. So for example, we want to look at our macrophages, so we'll draw a box around our macrophages. And then we'll move those macrophages and we'll look at them more closely. So we do have a macrophage marker in here. It's that blue CD11B. And so what we'll see is we'll see a population that is negative for CD11B, and then we'll see our macrophage population that is positive for CD11B. So what we can say here is these are CD11B positive cells. And then over here, this is CD11B negative cells. All right, and then we can look at our lymphocytes. So we can create another box around this population here. And then we can look at them. Now for our lymphocytes, we have a few things going on. We have our CD4 and CD8. So if we do CD8 by CD4, we will have a population here that is negative. And we'll have our CD8s, which are orange. We'll color them orange. They'll be up here. And we'll have our CD4s that are green, and they're going to be over here. Now in our negative, that's going to be, for example, our B cells will be there. So we'll draw a few pink cells in there because there can be other things too, because they're not CD8 or CD4 positive. Now we could have looked at this a different way. We could have said, well, we're interested in our B cells and our CD4 cells. So we'll put CD19 here, and we'll put CD4 here. And again, we'll have our negative population here. We'll have our CD19 positive population here. And we'll have, again, our CD4s over here. And within that negative population, we're going to have our CD8s, of course. And so you can do many things with this. You can look at up to 23 different markers on these cells. You can ask, what type of CD4 cell do I have? What type of CD8 cell do I have? What type of macrophage do I have? This is, as I said before, the workhorse of immunology, because you can take cells out of a patient who is sick, cells out of a patient who is healthy, look at their populations of lymphocytes, and say, how are they different? And so it's very, very strong and important um, uh, assay for immunology. And so if you ever read immunology papers, you will likely see flow cytometry in some aspect of those papers. Now, another approach for diagnosis that is relatively new is using microspheres. And so you can use a Luminex. That is an assay that is commonly done with microspheres. Now, the technology is going to use ELISA and flow cytometry kind of together. And so you basically have polystyrene microspheres that are going to be color-coded with two different fluorescent dyes. And they can be identified using lasers similar to a flow cytometry. So you can have different dyes, and this means that each bead in a set of up to 100 can be given unique identity, which is referred to as a spectral symbol. So you can have 100 beads labeled with unique identity. And then this can be detected by a flow cytometer. Each bead can be coated with different compounds such as antibodies, enzymes, and they can collect different molecules from a test sample. And so it's going to be similar to an ELISA, where you have different colored fluorescent reporter tags, and the amount of a compound, such as antibodies, can be measured with the laser. The microspheres are going to be in a solution, and multiple beads are going to be in a single container, and each bead is going to have a different reactant. So you can combine up to 100 different compounds with a set of 100 beads. Keep in mind that the flow cytometry we just talked about, we're only up to about 23 different colors. So microspheres allow us to look at 100 different things at once. So you can do multiple testing. You can do diagnostic testing. You can look at different proteins in somebody's serum. And there are many things you can do to look at this, but it's all basically the same approach. So in this case, though, you have beads. So you'll have a microbead 1, microbead 2, microbead 3. And these will be tethered to some surface. They're usually going to be like a plate, a well, similar to what was used for like an ELISA. And then attached to each of them, you're going to have a different target. So one might have a triangle, the other one might have a square, and the other one might have a blue circle. And each of these targets is something that you're interested in seeing if an individual has a response to. So for example, this red triangle might be hepatitis A, and you can have the purple be hepatitis B, and you can have the circle be hepatitis C. And so, and then again, you expose your patient's serum, and they might have purple antibodies, for example. That will bind to hepatitis B, but their purple antibodies will not bind to hepatitis C, and it will not bind to hepatitis A. But in one well, you can detect if this person has antibodies to hepatitis. You can detect if this person has antibodies to hepatitis, HIV, tuberculosis, whatever you want to do. You can, as long as you have a bee that has the specific antigen that you're looking for, you can test a lot in a single go. And so this is a lot of power in order to be able to detect or use a small sample size, such as a person's blood sample, and look at many different specificities at the same time. Okay, so that's all I want to talk about with respect to antibodies, antigen interactions, and how we use them in the lab in diagnosis as well.